Okay, since everybody's here, I'm going to get started. So tomorrow's lab, okay? New lab tomorrow, exercise number 2. In exercise number 2, basically you're going to learn how to do some of the fundamental aseptic technique that allows you to start transferring things from one sterile environment to another. And this is going to be absolutely essential for lab next week, because next week you will learn how to do a couple of isolation techniques, streak plate and pour plate, and then you're going to get your unknown. Okay, so it's going to be important Friday to learn the techniques so that once you've got your unknown, you can subculture it and transfer it to new growth media and transfer it to media to do various tests and things. So it's really important to read the lab prior to coming to class tomorrow on Tuesday. Since we only have 15 minutes, there won't be a whole lot of time. And Dr. Morgan and I are going to try to get around as best we can to kind of make sure that your technique is acceptable and you're doing things the way you should be doing them. Okay? A couple of words. I've had a couple of questions about lab and documentation and notebooks and things like that. You're not going to have a traditional lab book. Actually, pretty much everything we do in terms of documentation will be through photos. And so you should get in the habit of bringing your device. Most of you are going to use your phones anyway for documentation. But when you come to lab, you should say, do I have something to take photos with? I do have an iPad here you can use if you need to. But here's what we're going to do, and I'll say a little bit more about this when we give you your unknown. You're going to collect all of your results from your unknown and document them with photos. And you will turn in your unknown report as a PowerPoint, okay? And it'll have all your results with it. There'll be some narrative as well, but it's mainly photo documentation of all of your test results. As you go through various labs, sometimes there are things that you're asked to do. For example, when you did the clean countertop, clean hands, dirty hands, it asks you to then evaluate your results once they're ready. We might have our plates ready Friday. I would probably guess beginning of next week it's going to take until we get some sufficient growth to do some good comparison. But in those particular situations, I'm going to ask you to photo document as well. And if there are things that need to be written, what you can do is we will have kind of almost 2 things you'll turn in at the end of the year. You'll have a kind of PowerPoint lab notebook, and then you'll have a PowerPoint unknown. Okay? And if you need to do anything text-wise, you can just put that into a PowerPoint slide. This is the PowerPoint lab notebook is a little bit new for me. And one of the reasons I'm going to have you do that is when we do labs, especially when we start doing tests on your unknown, we always have various known organisms as controls to compare your unknown to. And the tendency in the past has been for people to kind of gloss over the controls and just quickly glance at them. But then when they start working on their unknown, they went, man, was my unknown really negative or positive? And all I've got is this vague notation that E. coli was positive and Bacillus subtilis was negative. So what I'm going to have you do is I'm going to have you photo document your known organisms too as part of your lab notebook. And that's actually going to be helpful for you, because in that way you will be able to go back and use those controls later on if you have a question about your unknown. So I'll flesh that out a little bit more. I'll probably put together a document that describes what has to be in your lab notebook PowerPoint and what has to be then in your unknown PowerPoint as well for your report. But I just wanted to give you a heads up about that's kind of how we're going to be operating. So not a traditional lab notebook. And the questions at the end of each lab, if there are review questions, those are for your study guides basically for the lab practical. And so I'm not going to collect those and grade them, but I would encourage you to do those on a regular basis. And if you have questions about them, just ask, okay? If there's something that you don't understand or you want to make sure that you are answering questions correctly. Okay? So tomorrow's some lab work. Anybody have any questions? Okay, so we are in the process of talking about some bacterial cell structures that we will encounter in various microorganisms that we use in lab throughout the course of the semester, including your ANOA. And we're talking right now about the cell envelope. The cell envelope includes the cell membrane and the cell wall. And we're going to spend a fair amount of time talking about the cell wall because, number one, it's important structurally and functionally to the bacterial cell. It's really important to have that rigid cell wall around bacterial cells to protect them from swelling and lysing in hypotonic environments. But also, it's really important because cell wall structure is directly related to the outcome of the Gram reaction. And Gram staining is something that we're going to be doing in lab. And the results of a Gram stain are critically important in terms of identifying organisms and start splitting them out into groups when you use a dichotomous key. So Gram staining, cell wall structure is really pretty important stuff. With a couple of exceptions that we'll talk about in a little bit, all bacterial cells have some type of a cell wall. The vast majority of them have a cell wall made out of peptidoglycan. And we'll talk a little bit about what peptidoglycan is and how, for example, that differs from cellulose that you find in a plant cell wall. But the really important thing to understand right now is those bacterial cells that have a peptidoglycan cell wall fall into two categories, either gram-positive bacteria or gram-negative bacteria. And we looked at that a little bit at the end of last period. And this particular slide represents the arrangement of the gram-positive cell wall structure, which basically is a big, thick layer of peptidoglycan external to the cell membrane. And then the gram-negative cell wall structure is quite a bit different. Okay? It does have peptidoglycan, but a much thinner layer. What's unique about the gram-negative cell wall structure is that there's literally a second cell membrane that is part of that gram-negative cell wall structure. And we call that second cell membrane the LPS layer, the lipopolysaccharide layer. So here's a good slide to kind of start differentiating between gram-positive and gram-negative cell wall structure. So gram-positive cell wall structure on your left, gram-negative cell wall structure on your right. And it's nicely color-coded, and so the coding shows you some of the important molecules that are found in these cell wall structures. Immediately, if you start looking at this, you can see the brown, which is peptidoglycan, is much more extensive and thick layered in the gram-positive cell wall structure than in the gram-negative cell wall structure. Typically, about 90% of the mass in a gram-positive cell wall is made of that stuff called peptidoglycan. In gram-negative, much smaller amount of peptidoglycan, typically somewhere between 15 to 20% of the cell wall mass is made up of peptidoglycan in the gram-negative. In the gram-positive cell wall structure, that layer of peptidoglycan is pretty tightly pressed against the cell membrane. I disagree with this slide a little bit, and some of the textbooks that I use also disagree with it. Some agree with it. And that's in regard to this thing called a periplasmic space. I'm going to come back to that in a second, but in this particular slide, they're indicating that there's a very narrow periplasmic space between the peptidoglycan layer and the cell membrane. What we're going to see is that in a gram-negative cell wall structure, there is a very extensive periplasmic space. Many, probably most textbooks don't even use the term periplasmic space associated with gram-positive cell wall structure. They basically consider there to be no periplasmic space. They say that's really only a feature of gram-negative. So there's some disagreement there, but it's not a big point. But I wanted to point that out. One of the other things that you will see associated with the gram-positive cell wall structure are these red molecules right here. They are called teichoic acid, and there are some lipid derivatives of teichoic acid called lipoteichoic acid. You only find these molecules in gram-positive cell wall structure. That's really the only place we've ever found them. So they're only found in bacteria, and specifically only found in gram-positive cell wall structure. Teichoic acid is a polymer of phosphate and carbohydrate. And at physiological pH, phosphate groups carry a net negative charge. And so these particular teichoic acid molecules impart a net negative charge to the surface of a gram-positive cell wall structure. Now, there are going to be other molecules in the gram-negative cell wall that also contribute to a net overall negative charge to that cell wall structure as well. Why is that important? Well, when we start doing some staining reactions to try to visualize your cells better, you looked at some stained bacteria in lab yesterday, the Bacillus nigricans, and they were purple because they were stained with a purple dye called crystal violet. Crystal violet carries a net positive charge. And so one of the reasons that those bacterial cells stained with the crystal violet and retained that stain is because the positive charge of the stain was attracted to negative charges on the surface of the bacterial cell. And in the gram-positive case, which the Staphylococcus is gram-positive, most of those negative charges were the result of these teichoic acid molecules. There are some proteins that are associated with this thick peptidoglycan layer, but the amount of protein that's present is pretty minimal. But some of those proteins are toxic to other types of cells. For example, you've probably all heard of Streptococcus before. You might not have heard of the species of Streptococcus, Streptococcus pyogenes. It's a pretty nasty strep bacteria that can cause several different kinds of diseases. One of the reasons that these particular species of bacteria cause disease is some of their cell wall proteins that are associated with the peptidoglycan layer are cytotoxic molecules to other cells. And so if you get a strep infection, those strep cells can start killing your cells. I showed you a slide when we did the introduction of necrotizing fasciitis, flesh-eating bacteria. And one of the reasons that those particular strep bacteria can erode away skin tissue and basically cause the skin to fall apart is the toxic nature of some of those cell wall proteins that are interspersed in this peptidoglycan layer. But for the most part, the cell wall structure is a big old layer of peptidoglycan. Okay? Now, with the gram-negative cell wall structure, it's a little more complicated. Okay, so if you look at the right over here, here is our cell membrane. Here is our thin layer of peptidoglycan. 15 to 20% of the mass is peptidoglycan. And then external to the peptidoglycan layer is this complex second membrane, this LPS layer, this lipopolysaccharide layer. It's a phospholipid bilayer, really like the cell membrane is. But interspersed into that second membrane are these molecules of lipopolysaccharide. You don't find those normally in a cell membrane. So these are unique to the outer membrane, the LPS layer of the gram-negative bacteria. Those LPS molecules can be important. One of the lipids that makes up the LPS layer is a lipid called lipo-A. And lipid A is a molecule that is called an endotoxin. We'll talk more about endotoxins and exotoxins later on. But this molecule can be poisonous to other cells. Now, if the lipid A is part of an LPS molecule, then typically it doesn't cause a problem. But if you get infected with a gram-negative bacteria, and that bacteria dies, and that LPS layer starts to fall apart, and the lipid A is then liberated, then that can cause problems for your cells. Okay? Anybody ever had food poisoning before? Okay? You can have food poisoning from eating contaminated food. And there are a lot of bacteria that can cause food poisoning, but one of them is a gram-negative bacteria belonging to the genus Salmonella. Okay, so there's Salmonella food poisoning. Why is that making you sick? Because when those Salmonella cells die, that lipid A is being liberated from their LPS layer, and it particularly has an effect on intestinal cells and starts causing permeability changes in those intestinal cells, and that results in some of the nasty symptoms associated with food poisoning: diarrhea, nausea, vomiting. This particular disease typically is not fatal unless you start losing a whole lot of fluid as a result of untreated diarrhea, for example. That can cause you to go into hypovolemic shock, which is pretty serious. But it can make you pretty sick, okay? So endotoxins typically aren't lethal. Later on, we're going to talk about some other molecules that are toxins called exotoxins, which really are very lethal in cases. So we'll come back and talk about these endotoxins. But I wanted to introduce that to you right now and give you an idea of where these molecules come from. There is a pretty extensive periplasmic space in this gram-negative cell wall structure. And the periplasmic space is considered to be the space between the regular cell membrane and the LPS layer. So peptidoglycan would be considered to be part of that periplasmic space. Periplasmic space is kind of interesting. There's a lot of metabolic reactions that can happen in the periplasmic space of a gram-negative bacteria. And many of those metabolic reactions involve enzymes. One of the things that this second membrane does is it keeps those enzymes localized in that periplasmic space and keeps them from getting away where they're needed. Some of those enzymes are involved in, for example, external digestion of molecules that the bacteria might be eating. So there might be amylase out here in this periplasmic space. And that's helping to digest carbohydrate, starch, that would be in the environment. So the periplasmic space is kind of a localized place where some interesting things happen. This LPS layer is a selectively permeable membrane, just like the cell membrane is. Embedded in that LPS layer are a number of molecules that are proteins that are called porins. And we've identified over 30 of those porins that are located in the LPS layer. And those primarily serve a function of transport proteins. So they can bring things across that membrane that the membrane normally would be impermeable to. Now, that has some implications medically. We mentioned penicillin the other day. The naturally occurring form of penicillin that's produced by the fungi, Penicillium notatum, is very effective against gram-positive bacteria, but almost totally ineffective against gram-negative bacteria. Now, do you remember what penicillin does? What's the mode of action of penicillin? Okay, it doesn't really degrade it. It prevents its resynthesis of the cell wall, okay? And that seems like kind of a minor point, but it's actually fairly important. So remember, as the bacteria is weakening its cell wall to accommodate growth in the presence of penicillin, it can't resynthesize it again, so the cell wall effectively does get weaker, okay? So why does penicillin, naturally occurring form of penicillin, work well against gram-positive, but lousy, if not at all, against gram-negative? They both have peptidoglycan, right? It could be affected.
Maybe the proteins have something to do with the resynthesis.
Well, in both cases, the penicillins are inhibiting enzymes involved in the resynthesis of the cell wall. But why would these be pretty much unaffected? Why would the peptidoglycan? It can't get through the outer LPS layer. Okay, so that LPS layer is impermeable to the naturally occurring form of penicillin. So what have clever biochemists done? They've chemically modified naturally occurring penicillin to form some synthetic forms of penicillin, like amoxicillin. Probably all of you took that at some point in time when you were little. Probably in the form of a pink liquid for an ear infection. Okay. Semi-synthetic amoxicillin is slightly different and can be transported across that LPS layer, and therefore is effective against both gram-positive and gram-negative bacteria. Amoxicillin has a broader spectrum of action than naturally occurring penicillin does. It works against both gram-positive and gram-negative. Naturally occurring penicillin is primarily only narrow spectrum, only works against gram-positive bacteria. So there's some consequences to having this outer layer here in terms of what molecules can penetrate into the periplasmic space and ultimately can penetrate into the cytoplasm of the cell. So that is important. So here's a nice summary of comparison between gram-positive and gram-negative cell wall structure. Chemical composition, number of layers, thickness, etc. And again here, in this particular slide, they do say that there is a periplasmic space in the gram-positive, but it's very, very narrow, okay? Probably almost non-existent. Here, the periplasmic space, excuse me, is quite extensive. Well, a key component in both of those cell wall structures is this substance called peptidoglycan. So what's with this peptidoglycan? What is it? What kind of substance is it? Well, peptidoglycan is a polymer that consists of both sugars and amino acid derivatives, or modified amino acids. There are two types of sugars, NAG and NAM. NAG stands for N-acetylglucosamine. NAM stands for N-acetyluronic acid. So they're kind of glucose-like, but they're different than glucose. They contain some nitrogen, for example, that glucose doesn't. And so if you look over here, you can see that this is the structure of the NAG molecule, and this is the structure of the NAM molecule. You don't have to memorize those structures, okay? But you do need to know that there are these two different types of sugars. And what happens is that these sugars become covalently bonded to one another into chains that can be quite long in length. So an NAG alters with an NAM, another NAG, another NAM. But then additionally, one of these sugar chains can be attached to an adjacent sugar chain by this area in green down right here, which is the amino acid portion of peptidoglycan. This amino acid portion is referred to as the tetrapeptide portion. So up here in this particular representation, purple, yellow represent the sugars, covalently joined together to form a polymer, and then the green represents this tetrapeptide region that can bond one of those chains to an adjacent chain. So basically, peptidoglycan are huge, extensive sheets of these NAG, NAM, which are also called glycan or sugar molecules, joined together by these tetrapeptide linkages. So peptidoglycan is also called glycan tetrapeptide. Just another name for it. These particular amino acids are kind of interesting. There are some unusual amino acids found in this tetrapeptide region. There's a modified amino acid that I'm sure you've never heard of before called diaminopimelic acid. Why have you never heard of it before? Because we don't find it any place other than in prokaryotic peptidoglycan. So it's certainly not in your cells. And then there are some amino acids that you've heard of before. You've heard of alanine, glutamate, lysine, etc. But what do these designations over here to the left indicate? L and D, the organic chemists. Dextrorotatory and levorotatory, different optical isomers of those amino acids. If you look at your particular amino acids in your proteins that make up your body, one of those two stereoisomers is completely favored evolutionarily for some reason over the other. Which one?
D. Is it L?
L. Yep. Our proteins are made up of the L stereoisomer of amino acids. We do not find D amino acids in our protein structure. But you find them in prokaryotes, at least in this tetrapeptide region of peptidoglycan. How come? That's kind of interesting. Another interesting question is, why did evolution seem to favor that one stereoisomer to make polypeptides over the other? We don't know. There's some interesting speculations. So the bottom line is that these linkages right here are kind of unusual in terms of their chemical composition. The nature of these particular tetrapeptide linkages can vary from one type of bacterium to another, and particularly in gram-positive and gram-negative bacteria. So there are subtle differences. The number of these connections, how extensive there are, also can vary. Ultimately, the way these sugars are bound together to one another in these interacting chains ultimately determines the shape of the cell. Remember we said that cell walls determine the shape of cells? And if you remove the cell wall from a cell, it's round. But once you put a cell wall around it, then it can be a spirillum shape, or a bacillus shape, or a cocci shape. So basically, how these subunits are organized together, how extensive the linkage is between one of these chains to the next, the nature of the bonding and the types of amino acids that are occurring in the tetrapeptide, ultimately are determining bacterial cell morphology. So structure really does determine function. So, both gram-positive and gram-negative bacteria have peptidoglycan. Gram-positive has more, much more. Gram-negative bacteria additionally have this LPS layer that the gram-positive bacteria lack. So why does that translate into a different type of reaction from this staining procedure that we're going to do over and over again this semester until you're sick of it, the Gram reaction? If you've been through a microbiology lab, you better know how to do a Gram stain. One of the things that I will tell you as you start approaching studying for the lab exam is that I typically will not make you memorize staining procedures. How many seconds you do this, et cetera, et cetera, et cetera. Okay? With one exception, the Gram stain. This is something you will have to be able to do from memory. Okay? And I will tell you right now that on the first lab exam, there will be a set of 3 stations set up, right where Macy and Sunday and me are sitting, where you're going to be given a bunch of stains and a couple of test tubes of unknown bacteria, and you're going to have to do a Gram stain of that, and then report out both the Gram reaction and the cell morphology of your particular test. Okay. You're going to have to memorize it. And it's something that will just come naturally, because by the time you do these a few times, you will just know how to do the Gram stain. Okay? So, what is a Gram stain? Well, it's a type of stain that is called a differential stain. A differential stain allows you, based upon the outcomes of the stains, to separate bacteria into 2 separate groups. In this particular case, gram-positive and gram-negative. Not all stains are differential stains. Yesterday, we used a set of slides that involved a process called structural staining. So we'll differentiate a little bit more between structural and differential stain in lab. But Gram stain is a differential stain. Differential stains use 2 or more different staining reagents. Structural stains oftentimes just use a single staining reagent, like crystal violet, for example, when we looked at the stain slides of Bacillus and cocci. Okay, so over at the left, in the Gram staining procedure, shows you some of the staining reagents that you'll use in the Gram stain. We use a stain called crystal violet. That is a purple, positively charged stain. We use a substance called Gram's iodine. Gram's iodine is a mordant. I'll explain that in a second. We use 95% alcohol. And we use a red stain, a positively charged stain, safranin, which we'll see later is a counter stain. So these are our main staining reagents. And those are used in the order in which they are listed. So first you do crystal violet, then Gram's iodine, etc. I'm not going to get into how many seconds of each one of these you do. You'll do that in lab.
Why does the safranin has to be done if after step 3 you can already see a difference?
We'll get you there. Okay, so what happens during the Gram staining procedure? Well, the first step in the Gram staining procedure, applying the primary dye, crystal violet, doesn't give you any differentiation, because crystal violet will bind to the cell surface of both gram-positive and gram-negative cells, because both of them have negative charges on their surface. So at the end of the first step, both cells look the same. They're purple. The second step involves adding an iodine solution called Gram's iodine, and Gram's iodine interacts with the crystal violet and forms a complex with it. Has anybody ever heard the term mordant before? How about mortar? What is mortar? It's kind of a cement, isn't it? Something that holds bricks together, for example. So in this particular situation, the Gram's iodine is binding to or complexing with the crystal violet, kind of like mortar combines with bricks to hold them together. So what happens when you add the iodine to your cells that you previously treated with crystal violet? They're still both purple. And we still haven't gotten anywhere yet. Okay? The third step in the gram staining procedure is the most critical, and that is the application of alcohol to those cells. Alcohol is serving as a decolorizing agent. And what it does is it rinses the crystal violet iodine complex out of the gram-negative cells, but does not rinse them out of the gram-positive cells. So one of the questions becomes why? Well, what's the difference between gram-positive and gram-negative? Why can you decolorize these but not these? Now, you've got to be very careful to time this properly. Because if you decolorize for long enough, you can wash out the purple color of a gram-positive bacteria. So you can't decolorize too long. But if you decolorize for too short a period of time, you might not decolorize a gram-negative. Does that make sense? You really got to get the timing particularly correct. Now, Archer asks a very good question. Why can't we stop the gram staining procedure right here? Any ideas? It's pretty easy to see these purple stained cells, right?
Is it because, like, if you do end up doing it too long, the gram-positive cells would be white, but they won't pick up the stuff?
Well, one of the things we're going to talk about and really demand that you do is when you do a gram stain, pretty much on each individual slide that we do, if you're going to look at a gram stain of your unknown, then what we're going to be asking you to do is to put a control on the same slide, both of gram-positive and gram-negative. So that way, if the gram-positive cell after the decolorizing and gram staining is purple like it should be, and this one is pink like it should be, then you can be fairly confident that the results of your unknown are the same. If you put them on 3 separate slides, then you can't be as sure. So that's kind of how we address this issue of decolorizing too long or too little. But what if we decolorize just right? What's the problem with this cell?
You can't see it.
You can't see it very well. Okay? So just a lack of contrast between that and its environment. So what do? We provide another red stain dye. What happens when we put a kind of a weakly red stain dye into an intensely purple cell? Still purple. Okay. It's a mixture of red and purple, but it's still purple. But a colorless cell picks up the red stain dye. And now we can clearly differentiate between the ones that have been decolorized and not. Okay? So, what's the real difference? The real difference ultimately has to do with the thickness of the peptidoglycan layer. And the fact that it is a much more extensive layer in the gram-positive than in the gram-negative. So basically, if you get your timing right, you can time it so that if you decolorize just long enough, you can wash all of that stain out of the thin peptidoglycan layer, but you won't have washed it all out of the thick peptidoglycan layer. So the different response you get in gram staining is ultimately a reflection of that difference in thickness of peptidoglycan layer. The LPS layer has a little bit of effect too, but primarily retention of the dye is a product of how thick that peptidoglycan layer is. Okay? So like I say, we will be doing gram staining over and over and over again. Well, I said the vast majority of bacteria have cell walls made up of peptidoglycan. But there are some exceptions / modifications to peptidoglycan cell wall structure. One slight variation is really kind of a modification of peptidoglycan structure. These bacteria do have peptidoglycan. But in addition, they have another fatty substance in pretty large amounts. They've got lots of this substance impregnated into the peptidoglycan, and this substance is called mycolic acid. Bacteria that have this mycolic acid in their peptidoglycan are referred to as being acid-fast. And it has to do with a stain called the acid-fast stain. They show a positive acid-fast stain. And we will do an acid-fast stain in lab and talk a little bit more about the chemistry of what's happening and what makes something acid-fast. But it's basically the presence of these fatty molecules that gives you this different appearance for acid-fast bacteria and non-acid-fast bacteria. So why mention this? Well, there are a couple of genera. One which I'm sure you've never heard of before, called nocardia. And another which you may have heard of before, Mycobacterium, which are both typically acid-fast. There are two species of Mycobacterium that have a lot of medical significance. One of the species is Mycobacterium tuberculosis, obviously causative agent of TB. And the other is Mycobacterium leprae, which is the causative agent of leprosy. Tuberculosis used to be just a scourge in this country and around the world. It's relatively easy to diagnose with a chest x-ray because of the way your body responds to tuberculosis infections. You form these structures called tubercles in your lungs, and they clearly show up in an x-ray. But what if you don't have access to an x-ray? Is there a quick and dirty way to kind of screen for tuberculosis? And the answer is yeah. Someone comes in with symptoms of TB, oftentimes a hacking cough, for example. And what can a doctor do? Get a sputum sample, spit up some junk from your lungs, and do an acid-fast stain. And if you get positive acid-fast bacillus, then immediately you're suspecting that they're either Mycobacterium or nocardia, and likely symptoms of respiratory infection and things would suggest Mycobacterium tuberculosis. So we will do an acid-fast stain as part of our kind of series of tests that we do to evaluate your unknown. Another exception to the typical peptidoglycan cell wall structure are the Archaebacterium. Now remember, these constitute one entire domain of the three-domain system of classification. So this is an extensive group of organisms. In Archaebacterium, the cell wall is not made of peptidoglycan. Sometimes it's made entirely of protein. In other Archaebacteria, it is made entirely of carbohydrate. There's a lot of variation in cell wall structure within the Archaebacteria. A third exception to the rule. There are a group of microorganisms, a bacteria called mycoplasms. And they entirely lack a cell wall of any type. Here's an electron micrograph of some mycoplasms. And one of the things that might strike you as you start looking at these particular bacteria is these are all the same species of bacteria. In fact, these are all probably genetically identical cells. They've got a lot of different shapes, don't they? Mycoplasmics are highly pleomorphic, meaning their cells can assume a number of different shapes. Why are mycoplasmics notoriously pleomorphic? Remember we said one thing the cell wall does, determine cell shape, right? You may have heard of mycoplasms from a medical standpoint. They can be responsible for a form of pneumonia that is sometimes commonly referred to as walking pneumonia or mycoplasmic pneumonia. What's a kind of risk that mycoplasms are going to encounter due to the lack of a cell wall? Yeah, lack of protection against swelling up and bursting. Okay? One of the ways they compensate for that a little bit is that we're going to see later on that the cell membranes of prokaryotic cells are a little bit different than the cell membranes of eukaryotic cells. One of the things that eukaryotic cells do is that they have a fair number of sterol molecules, including cholesterol, that's a functional part of their cell membrane. And sterol molecules tend to give some rigidity to cell membranes. Mycoplasms are unique in being prokaryotes in that they tend to have sterol molecules in their cell membranes. So it's almost kind of like an adaptation against not having a cell wall. And then finally, there are some very bizarre bacteria called L-forms. L-forms are bacteria that sometimes have a cell wall, and sometimes they don't. It's really kind of a product of where they are in their life cycle. Well, this could be really make it pretty hairy in terms of identification of these particular bacteria. Because things that lack a peptidoglycan cell wall typically end up staining as gram-negative. Now that kind of makes sense if you think about it. Because if you don't have peptidoglycan, there's nothing to hold that crystal violet iodine complex in there. So you would quickly decolorize it and it would appear to be gram-negative. Well, what if I gave you an unknown that was an L form? And I said, okay, do your gram stain. And when it had a cell wall, it had a gram-positive cell wall, and then it lost it. So what would happen? Sometimes it would be gram-positive, sometimes it would be gram-negative. That would really be hard for you. And fortunately, I can guarantee you that none of your bacteria fall into this category of that being an L form. They're pretty unusual. Out of time. So we will wrap up here in terms of cell wall structure, or cell structure on Monday, and then continue on into the next topic, which is bacterial classification. So, sterile aseptic technique tomorrow. Yeah.
Mycoplasms. So they're inception. So when they're fully formed, they stay that shape.
Well, actually, minor variations in osmotic concentration of their environment can cause cells to take up or lose water, and ultimately that will affect their shape. So they're all over the place.
Okay.
You know, 1 minute they could be one shape, and then if minor osmotic changes occur, they could be a different shape.
My orders,
All right, thank you.
Sure.
before I had for today.
I've been looking through like your own lecture notes that we put on Google, and there were like some things that like weren't mentioned in class. Are we still like responsible for like the little minute details?
Well, I mean, not the little minute details, but if there is a kind of a topic, for example,
Right.
that I didn't quite get, then I think you should probably be familiar with that. I think that really ultimately what that would do is, maybe as you were writing an essay question, if you could include something as elaboration, it might give you a little bit. But in terms of the basic information, I mean, it's what we cover in class. But that kind of extra stuff that I might not have gotten to or just skipped over or something can help you sometimes to round out a question or to give, that just little bit better answer.
Okay.
I'm sorry if that helps or not. I mean,
Yeah, just like there was like so many like little things, like especially like with the benefits of microorganisms,
Yeah.
of like mining and like all the little like I don't know, like specifically like with the metabolism of the microorganisms and how like the elements can be going from the reduced to the oxidized state and
Right, and again, I mean, if we're talking about like an essay question, I ask you an essay question about, compare some of the beneficial versus, typically I will expect, some of the big things to be answered that we covered in class as well. But what if you kind of skipped over one of those, or a couple of them even, but then you ended up including something that you remembered from the notes? You went, oh, I remember something about, so that it can kind of be a supplement or a replacement for some of that kind of stuff.
okay.
So I don't know if that helps or not, but no, you don't have to know every
Little
tiny piece of information.
Okay, because I feel
Yeah.
like I know like the overall like especially what we talked about in class. I feel like I really know like that type.
Yeah. And so some of this other stuff is just kind of to round out the picture.
Okay.
I mean, it's kind of like reading. It's kind of like reading a textbook. I mean, no one expects you to know every absolute piece of information in there, but some of the additional information just kind of rounds out the picture. And
Okay.
including that sometimes can, give you a little bump. But I can't think of a situation where I would ever say, this is something you have to need to know and I'm going to take points off for it, even though I didn't talk about it in class.
Okay.
Okay?
Yeah. Thank you.
Sure. So, I mean, kind of think of the lecture notes as kind of a very abridged book for the course.
Okay. All right. There was just like I remember something about like the link between. It talked about different medical conditions. I don't have the notes with me, but I might come and ask you about it
Okay.
because I was kind of confused with that part of it.
Sure.